For decades, we've basically conceptualized psychiatric treatment by looking at the traffic of the brain.
Right, the momentary signals.
Exactly. We think about serotonin, dopamine, norepinephrine, these neurotransmitters just firing across the synapse in a fraction of a millisecond.
Yeah, very rapid, immediate sort of system.
And we tend to view depression as like a temporary traffic jam or maybe a shortage of specific vehicles on the road. But what if we've been looking at the wrong level of the system entirely?
And that's a real question, isn't it?
Right. What if the problem is not the momentary traffic, but the fact that the roads themselves are literally physically crumbling?
And that is exactly the paradigm shift we're unpacking today.
Right.
We are moving way beyond the momentary messengers. We're looking at the actual molecules that build, dismantle, and reshape the physical scaffolding of the nervous system.
The structural stuff.
Exactly. We are exploring neurotrophic factors.
Brilliant. Well, welcome to this deep dive, everyone. To truly navigate the complexities we're about to explore, we need to anchor our discussion.
We definitely do.
So I want you, the listener, to hold on to this foundational concept for the entirety of our journey today. Here it is. Psychiatry is the study of how brain, mind, and experience interact, and how that process can go wrong.
Spot on. That is the core of it.
Yeah. And today, we're taking a highly microscopic lens to Chapter 1.10 of Kaplan and Sadok's comprehensive textbook of psychiatry.
It's a dense chapter, but an absolutely crucial one.
Oh, incredibly dense. We're going to journey from the biochemical survival mechanisms of single cells right up through the macro-level clinical manifestations of things like obesity and trauma.
And then finally into the clinical reasoning behind how our modern psychiatric treatments actually function at a structural level.
Right, because I think it's remarkably easy in clinical practice to fall into the trap of viewing the brain as a static fixed machine.
Oh, absolutely. Like a computer where the hardware is permanently set by your late teen.
Yeah, exactly. And from then on, you're just, running different software.
But the biological reality is far, far more fluid than that. The brain is this living, constantly remodeled ecosystem.
It's physically changing shape.
It is. It alters its density, its connectivity, entirely based on the demands placed upon it by your environment.
Which is mind-blowing.
Oh.
And I find it helpful to conceptualize this using a specific mental model.
Okay, let's hear it.
Imagine the brain, not as a computer, but as a massive, sprawling, bustling metropolis.
Right, a city.
Yeah. And traditionally, psychopharmacology focuses entirely on the daily commute.
The traffic.
Exactly. The neurotransmitters are the cars, the pedestrians, the traffic lights, just sending signals back and forth in a fraction of a second. But neurotrophic factors, they aren't the traffic.
No, they're not. They are the infrastructure crew.
Right. And that analogy operates on several levels of accuracy, doesn't it?
It really does. Because these systems, they act far less like transient messengers and much more like nurturers. They are the sculptors of the environment itself.
They're the city planners, the structural engineers, and crucially, which we'll get into a bit later, the demolition teams.
Yes, the demolition aspect is vital.
They're the forces determining exactly where new roads should be built to handle increased demand. Which old, crumbling bridges need to be reinforced with steel.
And which dilapidated, unused districts simply need to be condemned and torn down to the ground.
Exactly. Okay, let's unpack this a bit more, because just like a real metropolis, if you don't have an active, highly regulated infrastructure crew, the city will inevitably fall into disrepair.
It has to. The structural decay will eventually dictate the flow of the traffic itself.
Which is why we're studying the neurotrophin family today.
Right. We are looking at 4 primary actors that orchestrate this structural integrity.
Let's name them.
Sure. We have nerve growth factor, or NGF. Then there's brain-derived neurotrophic factor, universally known as BDNF.
The big one.
The very big one. And finally, neurotrophin 3, or NT3, and neurotrophin 4, NT4.
Okay, so let's track the life cycle of one of these workers. I mean, a steel beam doesn't just appear fully formed on a construction site, right?
No, of course not.
And neither do neurotrophins. The underlying biochemistry reveals a rather elaborate origin story here. They don't start out as active builders.
No, they're initially synthesized as precursors. We term these proneurotrophins.
Proneurotrophins, okay.
Yeah. And the journey from this quite bulky precursor to a mature active protein is a highly orchestrated process of precise cleavage.
Cleavage, meaning cutting.
Exactly. Essentially, molecular scissors must cut away specific parts of the precursor to fold it into its final active shape.
I'm trying to visualize this physically. We have this bulky, inactive protein just sitting inside the cell. It's essentially waiting on standby. Where exactly does this cutting, this cleavage take place?
Well, it can happen at multiple distinct stages, which is honestly part of the system's brilliance.
Oh, really? Not just in one place?
No, it can occur intracellularly, so deep within the Golgi apparatus of the neuron, facilitated by an enzyme called furin.
Furin.
Right. Or it can happen as the protein is being packaged within dense core vesicles via proconvertases.
Okay, so still inside the cell at that point.
Yes, but fascinatingly, it can also happen extracellularly.
Wait, outside the cell?
Precisely. After the inactive precursor has already been released outside, driven by extracellular enzymes like plasmin or specific matrix metalloproteinases.
Wow.
And once cleaved, the mature proteins are relatively small, around 12 to 14 kilodaltons, and they bind together to form these stable, non-covalent dimers.
Okay, but why build such a convoluted manufacturing pipeline? I mean, why not just have the cell produce the active, ready-to-go BDNF immediately when it's needed?
Because building massive proteins from scratch takes a significant amount of time.
And the brain needs to be fast.
Exactly. The brain often needs to adapt to environmental demands in minutes, not hours. So this multi-step creation and cleavage process provides the nervous system with multiple distinct points of regulation.
It's a stockpiling mechanism.
That's a great way to look at it. The neuron can stockpile these inactive proneurotrophins in vesicles right at the synapse. They are sitting there, locked and loaded.
Just waiting.
Just waiting. And they only become active when an environmental trigger like a burst of intense learning or physical activity causes the release of those specific cleavage enzymes.
It's a profoundly elegant system of resource management. You deploy the raw materials ahead of time, but you don't issue the building permit until the exact moment the activity demands it.
Yes, you've grasped it perfectly.
So they are incredibly responsive. And that responsiveness brings up another fascinating point about how these crews are deployed across their cerebral metropolis.
Right, their distribution.
Yeah, they aren't evenly distributed. The biological mapping shows a stark contrast in their territories. NGF, for instance, the very first neurotrophic factor we ever identified, it seems to operate in very specific, restricted districts.
It does. Its mandate is highly targeted. In the peripheral nervous system, NGF acts primarily on sympathetic neurons and sensory neurons involved in nociception.
Nociception, so the processing of pain signals.
Yes, pain and temperature sensation. But within the central nervous system, NGF is intensely focused on promoting the survival and functioning of a very specific subset of cells, the cholinergic neurons located in the basal forebrain.
And those basal forebrain cholinergic neurons, they project directly into the hippocampus, don't they? Making them absolutely vital for memory formation.
Precisely. The survival of those specific memory circuits is entirely dependent on a steady supply of NGF.
Okay, so that's NGF, very targeted. But contrast that restricted distribution with BDNF and NT3.
Great, it's quite a different picture.
Yeah, BDNF seems to be everywhere. It's heavily expressed across the entire cortex and the hippocampus. But honestly, that ubiquitous presence poses a logical problem for me.
Oh, so?
Well, if BDNF is everywhere and BDNF is a master builder, why isn't the brain just a runaway construction site? Why isn't it constantly building new synapses randomly all over the cortex?
That is the crux of how neuroplasticity actually operates. It is governed by a principle called activity-dependent plasticity.
Activity-dependent.
Right. BDNF does not build blindly. Its transcription, its release, and its cleavage from that inactive precursor state, they are all tightly coupled to neural activity.
So it only goes where the action is.
Exactly. We see massive surges in BDNF concentrations following physical injury, during intense neural stimulation after environmental stress, and particularly following physical exercise.
The system is inherently responsive. BDNF builds precisely where the structural reinforcement is demanded by the traffic.
What's fascinating here is that the electrical firing of the neuron is literally the signal calling the infrastructure crew to that specific street corner.
That's incredible. But the cellular machinery required to actually receive that signal and begin construction, that's extraordinarily complex, isn't it?
Highly complex. A molecule of BDNF floating in the extracellular space cannot accomplish anything unless it can dock with the cell. It needs a receiver.
Which brings us to the dual receptor system. And honestly, this is where the biological narrative takes a rather dark turn.
It does get a bit ruthless.
Yeah. We don't just have one type of receptor for these neurotrophins. We have two completely distinct systems. One builds and the other destroys. Let's start with the builders. The tropomyosin-related kinase receptors, or the TRK family?
The TRK receptors are the quintessential survival and growth receivers. They belong to a family called receptor tyrosine kinases.
Try saying that 3 times fast.
Right. Physically, you can picture them as having an antenna protruding outside the cell membrane to catch the neurotrophin.
Okay, an antenna.
Then there's a single column crossing through the cell wall and an intracellular domain, the tyrosine kinase, hanging down inside the cell, just ready to trigger chemical reactions.
And there's a strict selectivity here, isn't there? Different members of the crew only talk to specific foremen.
Yes. They pair off quite specifically. NGF binds highly specifically to tiroparp.
Tutrea, got it.
BDNF and NT4, they bind to TRKB. And NT3 primarily binds to TRKC.
Okay, let's look under the hood of exactly what happens when BDNF, for example, finds a TRKB receptor. How do we get from a molecule docking on the outside of a cell to actual physical growth happening on the inside?
It's a massive chain reaction. When a mature neurotrophin dimer binds to the extracellular portion of the TRK receptors, it physically pulls 2 of these receptors together in the cell membrane.
Pulls them together.
Yes. This pairing is called dimerization. And once paired, their intracellular catalytic domains activate, and they autophosphorylate.
Autophosphorylation. I always visualize that like a system turning its own keys in the ignition. It's a self-activating protocol.
That is a very apt visualization. By attaching phosphate groups to its own intracellular tyrosine residues, the receptor creates highly specific glowing docking stations on the inside of the cell.
Like landing pads.
Exactly.
Right.
And these phosphorylated tyrosines attract internal adapter proteins. The most critical adapter protein in this process is called SHUTLU. Right. You can think of Sherati as the master switchboard operator. Once Sherat docks to the activated TRX receptor, it routes the signal down 2 entirely separate, massive biochemical cascades.
Okay, let's map out these 2 cascades because this is the fundamental machinery of a living brain. Cascade number 1 is the survival pathway. If a cell is constantly on the brink of death, how does this pathway save it?
Well, the Sherat adapter activates an enzyme called phosphatidylinositol 3 kinase.
Or PI3K for short, thankfully.
Yes, universally known as PI3K. This PI3K then activates another crucial protein kinase called ACT.
What is ACT actually doing down there in the cell?
ACT's primary job in this context is to act as a molecular brake on the cell's suicide machinery.
That's suicide machinery.
Yes, apoptosis. ACT actively phosphorylates and disables pro-apoptotic proteins. It is the ultimate survival signal. As long as ACT is active, the cell is chemically forbidden from dying.
Wow, so the PI3K ACT pathway is basically the foreman holding up a giant stop sign to the demolition crew.
Exactly that.
Now, what about the second cascade? The one responsible for actually changing the shape of the city.
This is the plasticity pathway. Here, the Sherenzi switchboard routes the signal to increase the activity of ROS and mitogen-activated protein kinase, or MAPK.
Okay.
This cascade dives deep into the nucleus of the cell. It fundamentally alters the neuron's transcriptional programming by inducing the cyclic adenosine monophosphate response element binding protein.
Which is the CREB transcription factor.
Yes, CREB.
That is a massive player in the biology of psychiatry.
Incredibly massive. CREB physically binds to the DNA inside the nucleus and unzips it to initiate the production of brand new structural proteins.
So it's drafting the blueprints.
Right. It drives neurite outgrowth and is absolutely essential for long-term synaptic plasticity. Furthermore, this second pathway also activates the mammalian target of rapamycin complex, or mTORC.
I want to pause on mTORC for a second because the underlying mechanism here is brilliant. If CREB is in the nucleus, the City Hall drafting blueprints, how does a distant synapse, maybe millimeters away down a dendrite, actually get the materials it needs quickly?
That is exactly the problem mTORC solves. mTORC regulates localized activity-dependent translation.
Localized translation.
Yes. It acts like a local 3D printer situated right at the dendritic spine. It takes pre-existing messenger RNA that has been shipped out from the nucleus, and it immediately translates it into structural proteins right there at the synapse.
No waiting for delivery from the nucleus.
Exactly. This allows the synapse to physically remodel itself and strengthen its connection, the process of long-term potentiation, or LTP, without having to wait for new materials to be shipped all the way from the cell body.
So to pull the TREG system together, a mature BDNF molecule docks, the receptors pair up and turn their own keys. The Shale foreman takes the signal and splits it.
Right.
Half goes to ACT to say do not die. The other half goes to MAPK, CREB, and mTORC to say print new steel and build a stronger bridge. It is a profound structural empowerment of the neuron.
It is. But as you mentioned earlier, there is a darker side to this dual receptor system. We have mapped out the builders. Now we must address the P75 neurotrophin receptor, or P75-NTR.
If TRNK is the receptor tyrosine kinase, P75-NTR belongs to a completely different lineage.
A very different lineage.
It's a member of the tumor necrosis factor, or TNF, receptor superfamily. And while its outside antenna catches neurotrophins, its inside portion contains a protein association region that biologists have ominously named the death domain.
A very fitting name, unfortunately. While the TRK system is universally pro-survival, P75-NTR is a pro-apoptotic receptor. It is the demolition crew.
Right.
Its activation triggers an intracellular cascade of Cas-based enzymes that literally dismantle the cell from the inside out.
But this presents a massive biological contradiction to me. If neurotrophins can bind to both TRK and P75-NTR, how does the cell know whether it's supposed to build or self-destruct?
This brings us back to our discussion of the origin story. The cleavage process is the deciding factor here.
The cutting?
Yes. The uncleaved precursor forms, the proneurotrophins, have a very high affinity for the P75-NTR receptor. They preferentially bind to the death domain, but the mature, fully cleaved neurotrophins selectively bind to the TRK receptors to promote survival.
Let me see if I can translate this into our city metaphor. The uncleaved proneurotrophin is essentially an eviction notice. It gets sent out, binds to the P75-NTR mailbox, and tells the cellular building to demolish itself. But if that proneurotrophin encounters an enzyme, perhaps an extracellular protease released because that specific neural pathway is being heavily used, it gets snipped.
It gets cleaved.
Right. And that eviction notice is instantly transformed into a building permit. It now binds to TRK and demands structural growth.
That is an excellent way to translate it. The physical shape of the molecule dictates its destiny, and that shape is controlled by environmental activity.
But why would a developing nervous system even have eviction notices? I mean, why intentionally design a system capable of inducing its own cellular death?
Because efficiency demands ruthlessness. This is articulated in the neurotrophic hypothesis of development.
Okay, what does that dictate?
During early neurodevelopment, the body massively overproduces neurons. Imagine millions of axons all growing outward from the spinal cord, racing toward a single target tissue, like a specific muscle fiber.
Race.
A race where the muscle fiber is only capable of secreting a very limited, finite amount of nerve growth factor.
So it's a competition, a microscopic race for resources.
Exactly. A brutal competition. The neurons that successfully reach the target first and internalize enough of that limited NGF will trigger their CHK receptors.
And survive.
They will initiate that PI3K-ACT survival cascade we discussed, physically halting their own death. But the millions of neurons that arrive late or fail to secure enough NGF are left binding the excess proneurotrophins.
The uncleaved ones?
Right. Their P75-NTR receptors are activated, the death domain initiates apoptosis, and they are pruned away.
The winners get the retrograde transport of survival signals back to their cell bodies, and the losers are mercilessly dismantled to ensure the final neural network is perfectly tuned and highly efficient without any redundant wiring.
That's quite right. And what is truly remarkable is that this dynamic of reward and punishment does not stop when childhood ends.
It keeps going.
It continues into adulthood at the level of individual synapses, constantly shaping our active neuroplasticity.
Right. The clinical data shows us that mature BDNF evokes what is called chemical LTP. It is the biological equivalent of positive reinforcement, isn't it?
Yes, absolutely.
When a specific synaptic terminal is highly active, it releases enzymes that cleave pro-BDNF into mature BDNS right there in the synaptic cleft. That mature BDNF binds to TRKB, triggers the MTRC 3D printers, and stabilizes that specific connection. It rewards the active terminal.
Conversely, the inactive terminals are punished. If a terminal is not firing, it does not release those cleavage enzymes.
So it stays as pro-BDNF.
Exactly. The pro-BDNF remains uncleaved. It binds to the P75-NTR receptors on those quiet terminals, facilitating long-term depression, or LTD.
The opposite of LTP.
Right. The synapse literally receives a chemical signal telling it to retract, shrink, and dismantle its scaffolding. It is a constant dynamic balance of stabilizing what you use and pruning what you neglect.
We've spent a lot of time in the microscopic weeds here, looking at the nuts and bolts of receptors and kinases.
We have, but it's necessary.
It is. Because the reason this is so crucial is that this microscopic dance dictates our macro-level reality. When this infrastructure system alters, our behaviors, our bodies, and our mental health alter with it. Let's move into the clinical manifestations.
A very good pivot.
I want to start with metabolism and energy balance because I think most people intuitively link BDNF to mood or memory. But the textbook points out it is a master controller of our physical weight.
It is a phenomenal example of how integrated the brain and body are. BDNF and its TRKB receptor are absolutely central to the control of satiety, the feeling of being full.
Where is this happening?
We see this concentrated in a very specific region called the paraventricular hypothalamus, or the PVH.
The PVH. That's essentially the city's central metabolic control room.
Precisely. It is a cluster of nuclei containing distinct neuronal populations that express high levels of BDNF and TRKB. And the animal knockout models demonstrate just how powerful this system is.
What happens in the models?
Well, if you genetically delete BDNF specifically in the anterior PVH of a mouse, the clinical phenotype is staggering. The animals develop severe hyperphagia.
They completely lose the ability to stop eating.
Yes, extreme, relentless overeating leading to massive obesity. Conversely, if you inject recombinant BDNF directly back into the PVH of a control animal, it potently and immediately suppresses their food intake.
Wow.
And you see the exact same devastating hyperphagia if you delete the TRKB receptors in those feeding circuits.
But it's not just an intake problem, is it?
Yeah.
The absence of BDNF fundamentally alters the physics of their baseline energy expenditure.
That is what makes it a master controller. The dilution of BDNF in the PVH does not just make them ravenous, it drastically slows down their basal metabolism.
It drops their burn rate.
Exactly. Their baseline locomotor activity plummets. Furthermore, their thermoregulation is impaired.
Thermoregulation, so controlling body heat.
Right. These PVH neurons project down to the spinal cord to control the sympathetic networks that generate body heat. Without BDNF maintaining those specific descending pathways, the animals cannot effectively burn calories for warmth.
That's a double whammy.
It is, so they consume vastly more calories, and they burn significantly fewer.
And this isn't just an animal model anomaly, The source material explicitly connects this to human genetics.
It does.
There are rare missense mutations in the TRKB kinase domain or instances of BDNF haploinsufficiency that are directly linked to severe, early-onset hyperphagia and massive obesity in human patients.
It's quite striking.
It challenges you to completely reevaluate the divide between neurology and endocrinology. I mean, the exact same molecule that is currently rewiring your auditory cortex to understand this conversation is simultaneously holding up a structural stop sign in your hypothalamus to tell you that you don't need to eat right now.
It is the same underlying mechanism, facilitating synaptic transmission and structural integrity, but the clinical outcome depends entirely on the geographical location of the circuit. In the hippocampus, BDNF regulates memory. In the PVH, it regulates appetite.
Let's follow that logic into another circuit. Let's talk about the intersection of genetics, infrastructure, and fear.
A crucial topic.
The textbook details a very specific, heavily researched genetic variant, the Val66met polymorphism in the BDNF gene.
This is a cornerstone concept in psychiatric genetics. We are discussing a single nucleotide polymorphism, formally designated as RS62565.
Rolls right off the tongue.
Haha, yes. It represents a tiny substitution in the genetic code. A valine amino acid is replaced by a methionine amino acid at position 66.
But crucial to understand, this substitution doesn't happen in the mature functional part of the BDNF protein. It happens in the prodomain, the bulky precursor part that eventually gets cleaved off.
That's right.
Why would a change in the disposable packaging cause such a massive clinical problem?
Because that packaging dictates how the molecule is handled inside the factory. The Val66met substitution fundamentally alters the physical folding shape of the proneurotrophic.
So it folds wrong.
Yes. Because the shape is now slightly distorted, the cell's internal machinery, specifically the Golgi apparatus, struggles to sort and package it into the dense core secretory vesicles.
So the building materials are manufactured, but they get stuck in the warehouse. They can't be loaded onto the delivery trucks.
Exactly. The ultimate biological consequence is a significant decrease in the activity-dependent secretion of BDNF. The brain cannot release enough of the building material when it actually needs it.
And when is this most problematic?
Well, the animal models show us that this lack of secretion is particularly devastating during a highly specific critical window, the periadolescent period.
Adolescence. That's when the brain undergoes massive structural remodeling.
Yes, and specifically, the neural circuit connecting the prefrontal cortex to the hippocampus and amygdala is maturing. If a mouse or a human has the metal allele, That circuit does not receive enough BDNF during this critical window.
It's starved of infrastructure.
Right. Its structural maturation is permanently hindered.
And the clinical manifestation of that physically weakened circuit is a profound deficit in fear extinction.
Let's clarify exactly what cued fear extinction is, because it's really the foundation of how we treat anxiety and PTSD.
Fear extinction is the active biological process of learning that a previously dangerous cue is no longer a threat. It is not simply forgetting the fear, it is actively building a new inhibitory memory right over the old one.
It requires building new roads. Exactly. This requires massive synaptic plasticity in that prefrontal hippocampal circuit. If you have a patient carrying the BDNF metalleel, their physical infrastructure is impaired. They exhibit a prolonged, resilient response to conditioned aversive cues.
The brain quite literally struggles to update its physical scaffolding to unlearn the fear.
It does. And the textbook explicitly notes that in a clinical setting, a human carrying this metalleel would likely require a vastly different treatment approach.
How so?
They might need a much longer, more intensively tailored course of exposure therapy simply to achieve the same baseline extinction of a phobia or a trauma response as someone without the genetic variant.
This bridges the gap into gene-environment interactions. The text references meta-analyses demonstrating that the metalleel significantly moderates the relationship between stressful life events, particularly childhood adversity, and the subsequent development of clinical depression.
It fundamentally changes how we must view trauma.
Right. Trauma is not just a psychological event floating in the ether of the mind. It is a physical event that interacts with a genetic vulnerability to permanently alter the structural layout of your fear circuits.
Yes.
If you have the metalleel, your city's infrastructure crew is already understaffed. When the earthquake of childhood adversity hits, the city simply cannot rebuild the bridges fast enough, leaving you biologically vulnerable to mood disorders for the rest of your life.
That is a very precise integration of the biological mechanisms and the clinical reality. Before we focus entirely on major depressive disorder, we should briefly touch on how this infrastructure failure manifests in neurodegenerative and pain disorders, just to fully respect the breadth of the neurotrophin family.
Absolutely. Huntington's disease is a tragic textbook example of what happens when the cellular supply chain breaks down. Huntington's is characterized by the progressive death of medium spiny neurons in the striatum, deeply impacting motor control.
And the underlying mechanism directly involves BDNF. The striatal neurons do not produce their own BDNF. They rely entirely on BDNF being manufactured in the cortex and shipped down to them via anterograde axonal transport.
Shipped down the axon.
Yes. And the text points out that the mutant Huntington protein physically interferes with this transport mechanism.
The roads connecting the factory and the cortex to the consumers in the striatum are blocked.
Yes. The medium spiny neurons physically die because they are starved of the necessary neurotrophic survival signals.
It's devastating.
It is. Similarly, in Alzheimer's disease, we see the profound loss of those highly specific NGF-dependent cholinergic neurons in the basal forebrain that we discussed earlier. The infrastructure fails, and the memory circuits collapse.
And on the flip side, we have pain, where NGF is acting not as a savior, but as an amplifier.
Right. Pain is essentially a hypersensitization of the nervous system. After tissue damage or in chronic inflammatory conditions like osteoarthritis, there is a massive upregulation of NGF.
A surge of it.
Yes. This NGF binds to ChIR-KA receptors on the nociceptive sensory neurons, actively strengthening and sensitizing the pain pathways from the peripheral joint to the brain.
Which is exactly why the pharmaceutical industry developed neutralizing antibodies against NGF. By blocking the neurotrophin, you aren't just giving a painkiller, you are literally stopping the pain pathways from structurally reinforcing themselves.
Exactly. So, we see the pattern clearly here. Infrastructure failure in the PVH leads to massive obesity. Failure in the basal ganglia leads to Huntington chorea. Overactive infrastructure in sensory nerves leads to chronic pain.
Which brings us to the most profound paradigm shift in the textbook: integration and clinical reasoning in psychiatry. What does this physical scaffolding have to do with major depressive disorder?
This is where it all comes together.
Because for a long time, the public and clinical narrative has been dominated by the monoamine hypothesis. We tell patients depression is a chemical imbalance, a lack of serotonin traffic.
But the neurotrophic hypothesis does not necessarily replace the monoamine hypothesis, it vastly deepens it. It reframes depression, particularly stress-induced depression, as a fundamental dysregulation of synaptic plasticity and neuronal survival. Depression is a state of structural atrophy.
How does chronic stress physically cause that atrophy? What is the biological chain of events?
The preclinical data is robust. If you expose animal models to chronic restraint stress or unpredictable psychosocial stress, it leads directly to a sharp decrease in the expression of BDNF, specifically within the hippocampus.
So the survival signals drop.
Yes. Without that constant survival signal, we see actual physical retraction of dendritic arborizations. The branches of the neurons shrink.
Wow.
If the stress is chronic enough, it leads to the outright apoptosis, the death of hippocampal neurons, particularly in the CA3 subfield.
The city is actively decaying under the stress, and we see this exact same decay in humans, don't we?
We do. Human neuroimaging studies consistently demonstrate that patients with chronic major depressive disorder or severe PTSD exhibit a quantifiable volumetric decrease in the size of their hippocampus.
The physical structure of the brain has shrunk.
Yes. If structural remodeling and synaptic decay are the true cellular pathophysiology of depression, then BDNF is the prime candidate mediating these massive alterations.
Okay, I have to play devil's advocate here. I want to push back on this because it sounds almost too simple.
Fair enough. Go ahead.
If low BDNF in the hippocampus equals depression and the structure is decaying, why don't we just synthesize a massive batch of BDNF in a lab, put it in a pill, and give it to the patient? Just flood the brain with builder molecules. Why isn't BDNF the ultimate antidepressant?
That is the exact logical trap that early neuroscience fell into. And the textbook provides a critical warning against it. Context and circuitry are everything.
Context.
Yes, BDNF is not a generic happy chemical. It is a structural amplifier. It permanently strengthens whatever circuit it happens to be released in.
So what happens if you release it in the wrong circuit?
The textbook highlights a fascinating and counterintuitive example, the mesolimbic dopamine system.
Okay, the reward pathway.
Yes. This system involves the ventral tegmental area projecting to the nucleus accumbens. If you exogenously administer BDNF into this specific circuit, it actually increases depression-like and anxiety-like behaviors in animal models.
Wait, the exact same molecule that cures depression in the hippocampus causes depression in the reward circuitry. How is that logically possible?
Because in social defeat paradigms where an animal is subjected to chronic social stress, that mesolimbic dopamine circuit is mediating the feeling of defeat, submissiveness, and despair. If you add BDNF into that active circuit, you are not curing the depression. You are structurally reinforcing the neural pathways of despair. You are making the depression biologically permanent.
That's terrifying.
In fact, if you selectively genetically remove BDNF from that specific dopamine circuit, it creates an antidepressant effect.
That is a stunning level of circuit-specific complexity. It completely destroys the idea of a simple chemical imbalance.
It does, and it explains why indiscriminate flooding of the central nervous system with neurotrophins is a terrible, dangerous therapeutic strategy.
Even if you could get it into the brain.
Right. Even if you could get these massive 14 kilodalton proteins across the blood-brain barrier, which is incredibly difficult, flooding the brain leads to severe untoward side effects.
Because you're sending building permits to every single cell simultaneously.
Precisely. It induces widespread ectopic sprouting neurons making connections where they shouldn't. This can lead to severe epileptic activity.
Sure.
Yes. The brain becomes so overloaded with growth signals that the electrical grid misfires entirely. Furthermore, if you constantly bombard the TRKB receptors with BDNF, the cells protect themselves by heavily downregulating the expression of those receptors.
They pull the antennas inside the cell.
Exactly. The brain becomes deaf to the signal, which is exactly why direct BDNF application has shown minimal therapeutic effects in human clinical trials.
So we can't hand the patient a BDNF pill. That means our actual everyday psychiatric treatments, the pills we do prescribe, must be working through a backdoor mechanism.
They are.
How do traditional antidepressants like SSRIs and SNRIs actually interface with this infrastructure crew? Why do they take so incredibly long to work? I mean, every clinician knows it takes 10 to 20 days for a patient to feel the effects of an SSRI.
That 20-day timeline is the ultimate proof of the neurotrophic hypothesis. Traditional agents act by blocking the reuptake of serotonin or norepinephrine. This increases the momentary traffic in the synapse.
Right, the car's on the road.
Yes. These neurotransmitters bind to monoamine G protein-coupled receptors, or GPCRs.
But GPCRs don't build things directly, do they?
Exactly. They are just the first domino. Continuous daily activation of these GPCRs initiates a very slow-burning intracellular cascade. Over weeks, this constant signaling eventually upregulates the activity of our old friend, the CREB transcription factor.
CD Hall.
Yes. It takes time for CREB to accumulate in the nucleus. It takes time for CREB to bind to the DNA and induce the transcription of the BDNF gene.
It's a slow bureaucracy.
It is. It takes time for that new mRNA to be translated into proneurotrophins, packaged into vesicles, transported to the synapse, and cleaved into mature BDNF. And finally, it takes even more time for the brain to upregulate the expression of new TLKB receptors to actually catch this new BDNF.
Wow.
The extended timeline of therapeutic action perfectly parallels the biological timeline required to literally build new cellular infrastructure from scratch. You are not waiting for the serotonin to balance, you are waiting for the hippocampus to physically rebuild itself.
That makes the clinical reality make perfect biological sense.
Yeah.
But then, the textbook throws the most massive curveball in modern psychiatry into the mix: ketamine.
Ah, ketamine.
We know that subanesthetic doses of ketamine do not take weeks to work. They can alleviate severe, treatment-resistant depression and suicidal ideation in a matter of hours. If building infrastructure takes 20 days, how on earth is ketamine bypassing the entire construction timeline?
Ketamine is revolutionary because it acts through a completely different, vastly accelerated molecular pathway to achieve the exact same neurotrophic end goal. Ketamine is a fast-acting, non-competitive antagonist at the N-methyl-D-aspartate, or NMDA, glutamate receptor.
So it blocks a specific type of traffic light.
Yes, specifically on inhibitory GABAergic interneurons. By blocking these NMDA receptors, ketamine triggers a massive cascade. But crucially, the text notes a highly specific intracellular mechanism. This NMDA blockade leads directly to the deactivation of an enzyme called eukaryotic elongation factor 2 kinase.
EEF2 kinase.
We call it EEF2 kinase, yeah.
What does EEF2 kinase do in the normal depressed brain?
In a state of chronic stress, EEF2 kinase acts as a heavy parking brake on the ribosome, the cell's protein manufacturing plant.
Okay, a parking brake.
The neuron might have BDNF blueprints mRNA sitting right there at the synapse, but EEF2 kinase prevents the ribosome from actually reading them and manufacturing the protein.
It's an active blockade on construction.
Precisely. Ketamine rapidly deactivates EEF2 kinase. It instantly cuts at the brake line. The result is a massive, incredibly rapid synthesis of BDNF protein from those pre-existing blueprints within just 30 minutes of administration.
30.
Yes. This rapid surge of BDNF immediately binds to TRKB, activates the mTORC pathway we discussed earlier, and triggers immediate dendritic spine remodeling and synaptogenesis.
That is awe-inspiring. 30 minutes to rebuild the scaffolding versus 20 days. And the source text mentions a secondary mechanism for ketamine too, doesn't it? Something involving a glutamate burst.
Yes. By blocking those inhibitory NMDA receptors, ketamine also causes a rapid, transient burst of extracellular glutamate.
Which stimulates other things.
Right. This massive glutamate rush strongly stimulates a different type of receptor, the AMPA receptors. That intense, localized AMPA stimulation acts as a massive activity signal, directly causing the local release of stored, uncleaved neurotrophins, including both BDNF and vascular endothelial growth factor, or VEGF.
And VEGF is heavily involved in building new blood vessels, further supporting tissue growth.
Exactly. These released factors bind to their respective receptors, signal down through actin mTORC, and immediately promote the translation of necessary synaptic proteins. It is activity-dependent neuroplasticity thrown into absolute overdrive.
To round out our treatment discussion, we have to look at the oldest, most robust, and arguably most stigmatized treatment in psychiatry: electroconvulsive therapy.
ECT.
ECT is the gold standard for severe treatment-resistant depression. Are we literally shocking the neurotrophic system into gear?
The underlying premise holds true, though the clinical data is somewhat more nuanced than the animal models. In rodent models, a course of long-term electroconvulsive seizures robustly and undeniably increases BDNF transcription and elicits massive sprouting of new neurons in the hippocampus.
It forces the city to rebuild.
It does. In fact, if you induce seizures in mutant mice that are genetically engineered to have low BDNF, the therapeutic antidepressant-like effects of the ECT are significantly attenuated.
Meaning the seizure itself isn't the cure. The BDNF released by the seizure is the cure.
Correct.
But you mentioned the human data is nuanced. Why?
Because measuring this in living humans is incredibly difficult. We cannot biopsy a living human hippocampus to check BDNF levels. We have to rely on peripheral biomarkers drawing blood to check serum BDNF.
And what does the blood show?
The textbook points out that using peripheral BDNF levels to predict or track ECT treatment response in humans has yielded very mixed, inconsistent results.
Why such a discrepancy between a lab rat in a human patient?
Because human biology is chaotic. As we have discussed extensively today, human BDNF expression is subject to a massive variety of interacting factors. A human might have the Val66Met polymorphism. They might have a highly stressful home environment. They might have a poor diet or a lack of exercise.
Well, a lab rat lives in a highly controlled, genetically identical bubble.
Exactly. But despite the mixed peripheral biomarker data, the overarching biological theme remains robust. Whether it is SSRIs slowly ramping up CREB over weeks, ketamine rapidly cutting the brakes on EF2 kinase in minutes, or ECT electrically shocking the network into a massive growth response, all of our most potent psychiatric interventions ultimately rely on engaging the neurotrophic infrastructure.
It changes everything about how you explain a prescription to a patient. You aren't giving them a pill to balance their chemicals. You are giving them an agent that will help their brain physically rebuild its resilience.
That's a profound way to phrase it.
So, what does this all mean for the clinician, the trainee, or the curious listener? Let's distill the absolute key takeaways from this dense chapter. We will alternate through the most vital concepts you need to carry forward.
First and foremost, neurotrophic factors are not momentary messengers. They are the foundational molecules that actively support neuronal survival, growth, and long-term structural plasticity.
Second, BDNF is the master foreman of this process. It is the key molecule involved in synaptic formation, adaptation, and the physical strengthening of neural connections.
Third, because they literally govern the physical shape of the circuitry, these neurotrophic systems fundamentally influence how we learn, how we form memories, and how we adapt our long-term behaviors.
Fourth, and this is the core of the brain-mind experience interaction. Your environment directly controls this system. Enriched environments, learning, and physical exercise can actively enhance these pathways. The brain physically reacts to the life you lead.
The fifth takeaway is the tragic inverse. Chronic physical or psychosocial stress may severely impair these pathways. Stress causes physical atrophy in the hippocampus, reducing the neuroresilience required to bounce back from adversity.
Sixth, we must completely reframe psychiatric disorders. Major depression may fundamentally be a condition of reduced plasticity and structural decay, not just a simple chemical imbalance in the synapse.
And finally, our psychiatric treatments, from the slow onset of standard antidepressants to the rapid action of ketamine, may ultimately achieve their clinical efficacy not by fixing traffic, but by restoring this vital growth and adaptability within the neural infrastructure.
Excellent. Now, it is time for our learning reinforcement section. We have covered an immense amount of biochemistry and clinical reasoning today. We are going to test your retention.
A quick quiz.
Exactly. The expert speaker will read 6 single best answers style questions drawn directly from the mechanisms we just explored. I will insert a brief pause before the answer is revealed. I highly encourage you, the listener, to actively pause, think back to the specific pathways we discussed, and commit to an answer.
Let us begin. Question 1, focusing on mechanism. Which receptor, when bound by proneurotropins, acts as a pro-apoptotic signal during nervous system development? A, TRKA, B, TRKB, C, TRKCD, P75, NTR.
Think back to our discussion of the cellular demolition crew and the eviction notice.
The answer is D, P75, NTR. Remember, the uncleaved precursor forms preferentially bind to the P75 receptor to trigger the intracellular death domain cascade, forcing the cell into apoptosis during developmental pruning.
Right on.
Question 2, focusing on clinical reasoning. A patient with a BDNF Val66Met polymorphism undergoes significant childhood trauma. What specific psychiatric clinical manifestation are they most biologically vulnerable to due to impaired periadolescent circuit development? A, hyperphagia, B, impaired cued fear extinction, C, Huntington chorea, D, antipsychotic resistance.
Recall how that tiny shape change in the precursor packaging affects the specific circuitry connecting the prefrontal cortex to the amygdala.
The answer is B, impaired cued fear extinction. The MET allele hinders the normal packaging and secretion of BDNF, physically depriving the fear regulation circuits of the building materials they need to structurally learn that a cue is safe.
That's a crucial one for PTSD.
Question 3, focusing on concept. In the paraventricular hypothalamus, what is the primary direct consequence of BDNF or TRKB selective deletion? A, decreased nociception, B, increased energy expenditure, C, severe hyperphagia and massive weight gain, D, early visual cortex critical period termination.
Think about the intersection of neuroscience and endocrinology in the brain's master metabolic control room.
The answer is C, severe hyperphagia and massive weight gain. Loss of BDNF signaling in the PVH entirely removes the structural suppression of appetite and dramatically lowers baseline basal metabolism.
It's halfway there.
Question 4, focusing on mechanism. Traditional SSRIs require 10 to 20 days to demonstrate clinical therapeutic efficacy. In the context of neurotrophic pathways, this extended timeline directly correlates with the slow upregulation of which specific transcription factor? A, mTORC, B, EEF2 kinase, C, CRB, VEGF.
Remember the slow, steady process of sending blueprints to City Hall via G protein-coupled receptors.
The answer is C, CREB. Prolonged monoamine signaling slowly allows the CAMP response element binding protein to accumulate in the nucleus, unzipping the DNA to finally induce new BDNF gene expression.
City Hall at work.
Question 5, focusing on clinical reasoning. Why is indiscriminately flooding the central nervous system with exogenous BDNF considered a remarkably poor and dangerous therapeutic strategy for major depressive disorder? A, it rapidly degrades into toxic pro-BDNF. B, it induces profound, untreatable hyperphagia. C, causes widespread TRKB downregulation and potential severe epileptic activity. It prevents ocular dominance column formation.
Consider the systemic risks of activating every single growth pathway in the cerebral metropolis simultaneously.
The answer is C. It causes TRKB downregulation and potential epileptic activity. Broad, unregulated application leads to ectopic sprouting, massive electrical misfiring seizures, and forces the cells to pull their receptors inward to protect themselves.
And the final question.
Question 6, focusing on mechanism. How does the administration of ketamine bypass the typical multi-week timeline to rapidly initiate BDNF protein synthesis within just 30 minutes? A, by directly phosphorylating the intracellular domain of TRKB, by rapidly deactivating eukaryotic elongation factor 2 kinase via NMDA receptor blockade. C, by acting as a highly selective agonist at the P75 NTR death domain by actively blocking the extracellular cleavage of pro-BDNF by plasmin.
Think about how ketamine interacts with the ribosome's parking brake.
The answer is B, by deactivating EEF2 kinase via NMDA blockade. By cutting this brake line, ketamine instantly frees up the cellular machinery to begin translating pre-existing BDNF blueprints into active protein without waiting for nuclear transcription.
Fantastic. If you found yourself struggling with the biochemical specifics of those cascades, I highly recommend opening the textbook and reviewing the specific subsections in Chapter 1.10. Building these mental models requires repetition.
Before we conclude our exploration today, I want to leave you with a final provocative thought to ponder.
I love these.
We have established that neuroplasticity is an active, ongoing process. If our daily lived experiences, our chronic stressors, our physical traumas, and our persistent learning are constantly, relentlessly reshaping our neurotrophic landscape down to the molecular level, we must ask ourselves a profound question. How much of what we consider our fixed personality or our innate psychological traits is simply the structural, physical legacy of our past environments permanently etched into our brain's infrastructure by BDNF?
That is a staggering thought to end on. It truly blurs the line between the biological scaffolding and the psychological self. Thank you for joining us as we map the architecture of the mind. Here are the details of this episode.
Neurotrophic factors. Byline. The forces of growth and repair, how the brain sustains, adapts, and reshapes itself.